Hello everyone. Today we are solving Cambridge International AS and A level Biology Paper 2 Structured Question October-November 2021 Paper 2-3. Question number 1. Figure 1.1 is a diagram showing a stage in protein synthesis. So we can see a stage in protein synthesis. All right, we can see the molecule B, which is tRNA molecule leaving the ribosome. Another molecule D, tRNA, and another tRNA is bringing in the amino acid. Okay, and we can see the polypeptide, which is represented by C. Name the stage of protein synthesis that is shown in figure 1.1. So we are actually looking at translation. The question says identify A, B, C in figure 1.1. So A is actually messenger RNA. B is transfer RNA. And C is basically the polypeptide. State the base sequence at D and E. So at D is opposite to U, U, U. So it should be A, A, A. And at E, it is opposite to CAC. So C will be G, opposite to A will be U, and then G. So G, U, G. Part B. Mutagenesis is a process that leads to the change in the amino acid sequence of proteins. Scientists carry out mutagenesis to investigate the importance of particular amino acid in the protein structure and function. Outline how changing one amino acid in the beta polypeptide of hemoglobin may change the structure and function of the molecule of hemoglobin. So basically, if an amino acid is changed, different amino acid will have a different R group. So if one of the amino acid is changed, then it may prevent the formation of a certain bonds between the R group, and it can also change the secondary, tertiary, and quaternary structure, because the interaction between alpha globin and beta globin, all right, it can also change because of the interaction between the R groups. The change in one amino acid may change the ability of the hemoglobin to bind to oxygen and carbon dioxide. But question number 2, pepsin is an enzyme that hydrolyzes protein protease. Some students used pepsin from the stomach of a mammal. Okay. The activity of pepsin was investigated by placing a small quantity of the enzyme with a known concentration of the protein albumin. Figure 2.1 shows the progress of the enzyme catalyzed reaction that was carried out at 20 degrees Celsius. So we can see as the time increases, the concentration of the product, which is the amino acid, also increases. Calculate the initial rate of the reaction. So to calculate the initial rate of the reaction, we can start at the very beginning. We can see within 10 minutes, there was a total amount of product that was produced of 6 micromole per dm cube. So we can take this as our baseline. So we can use 6 micromole per dm cube divided by 10 minutes. And as a result, we're going to get 0.6 micromole per dm cube per minute. Part B1. The procedure was repeated to find the effects on the activity of pepsin using a competitive inhibitor at the same temperature. We know that generally when we add a competitive inhibitor will basically fight for the same space of active site. Competitive inhibitor occupies the same space as the substrate occupies in the enzyme. Predict the results that will be obtained using the competitive inhibitor. So the initial rate of the reaction will be lower, and it will take longer to reach 10 micromole per dm cube. The final concentration of the product will, however, still be the same. Part B2. The procedure was repeated without the competitive inhibitor but at a higher temperature of 30 degrees C. Predict the results that will be obtained at 30 C. So, if we are doing the experiment, and if we're repeating the experiment instead of 20 degrees Celsius at 30 degrees Celsius, the initial rate of the experiment will definitely be higher because the substrate molecules have more energy. All right. However, the final concentration of the product will be still the same. Part C. The student extended their investigation by using pepsin from a different species of mammal. The experiment was carried out at 20 degrees Celsius without a competitive inhibitor. With reference to figure 2.1, explain the advantage of calculating the initial rate of the reaction in each experiment. Actually, the initial rate of the experiment allows us to make a valid comparison between the activities of the enzyme. And also the initial rate is the fastest rate. It is not limited by the substrate concentration, because in the beginning, the substrate concentration is as perfect as we have provided. But as the reaction continues, the substrate concentration decreases. And it is also not influenced by the product that is produced, meaning that there is no end product inhibition. Question number 3. Figure 3.1 is a transmission electron micrograph showing red blood cells in a capillary of a healthy adult. So we can see the red blood cells. Explain how the cells in the capillary shown in figure 3.1 can be identified as red blood cells. Good question. We can see that these particular cells do not have any nucleus. All the other surrounding cells have nucleus. These particular cells do not have any nucleus. They're inside a tube. That's also we can see. The cytoplasm of that particular cell is very homogeneous. The cells have the same size as that of the lumen of the capillary. We can see that this is the lumen of the capillary, and the cells have the similar size, about 7 micrometer in diameter. The cells are flexible. We can see that the cells are squeezing through the capillary. Part A2. Capillaries are surrounded by tissue fluid. Outline the ways in which the composition of tissue fluid differs from that of blood. All right, we have done similar questions like this in our previous question paper. Tissue fluid does not have any red blood cell, and there will be no platelets in tissue fluid as well. All right, because they're too large to pass through the capillaries endothelial layer. Tissue fluid has less glucose, fatty acid, and amino acid. Tissue fluid has a low concentration of oxygen, because in blood the concentration of fatty acid, glucose, and amino acid is definitely going to be higher than that of tissue fluid, and oxygen concentration in blood is definitely going to be higher than that of tissue fluid, so that the concentration gradient is towards the tissue fluid all the time. Guys, one important advice: these questions are written with maximum number of marking scheme points as possible. It is advised that you try to write down these answers with the maximum number of points, so that when you learn these particular answers, even if you forget some of the points, you can still write a complete answer in your exam. Part B. Scientists compared features of physiology of 3 groups of people who live at different altitudes. These people were selected from populations that have lived at these altitudes for many thousands of years. Okay, scientists took blood samples from people at each location and measured the concentration of hemoglobin in the blood, the percentage saturation of hemoglobin with oxygen in the blood leaving the lungs, the oxygen concentration in the blood leaving the lungs. So we can see at near the sea level, Peru, mean hemoglobin concentration 15.3, mean percentage saturation 97, and mean oxygen concentration leaving the lungs. All right, mean oxygen concentration in blood leaving the lungs is 21.1 cm cube per 100 cm cube of blood. All right, and then the same information is repeated for 3,500 East Africa mountains. We can see mean hemoglobin 15.6, 95.5% saturation, same amount of oxygen leaving the, leaving in the blood that is leaving the lungs. Okay, and then from 3,750 to 4,000 in the Andes mountain, we can see that there is higher amount of hemoglobin. There is less saturation of that hemoglobin, and mean oxygen leaving the lungs is actually a little bit higher, which is 22. All right, now the question says, describe the changes in mean percentage saturation of hemoglobin with oxygen and the mean hemoglobin concentration in the blood as the altitude increases. So definitely we can conclude as the altitude increases, there is a increase in mean hemoglobin concentration in blood. It increases from 15.3 to 19.1 gram per 100 cm cube. All right, and then in terms of mean percentage saturation of hemoglobin with oxygen, we can see that mean percentage saturation of hemoglobin with oxygen as the altitude increases, we can see that it decreases. It goes from 97% to 92%. And then mean oxygen concentration in blood leaving the lungs, it, however, for 100 and 3,000 meter, it is same. But at 3,750 to 4,000 meter, it actually increases a little bit. So guys, this is a very good answer in terms of explaining this changes in mean percentage saturation of hemoglobin with oxygen, and then mean hemoglobin concentration in blood as altitude increases. Guys, moving to the next question. Part 2, with reference to table 3.1, such as how people living at high altitude can have an oxygen concentration in blood leaving the lungs similar to that people living at sea level. So the question says suggest. That means you have, a general understanding is that when a particular person is going to live at a high altitude, they're going to have more red blood cells, which explains why there is more hemoglobin in that particular, in the people of Andes, where there is more hemoglobin. It explains it very nicely. And then they may also have bigger lungs and chest cavity. As a result, they can take in more, oxygenated air into their lungs. They can have a higher cardiac output, means their heart can beat faster, and the release of erythropoietin hormone in their body can be higher, so which may stimulate more red blood cell production. Question 4. In tail crest Arabidopsis thaliana, most of the stomata are on the lower surface of the leaves. Okay, figure 4.1 is a diagram of an open stomata. Two guard cells are seen. Okay, some of the cellulose fibers in the cells of the guard cells are also shown. We can see the cellulose fibers are running in this direction. Okay, and we can also see the stomatal opening. Okay. The cellulose fibers shown in figure 4.1 are composed of bundles of cellulose microfibrils. Explain how molecules of cellulose are arranged into a microfibril. So basically, cellulose molecules have straight chains. They are arranged parallel. All right. So, when they're arranged parallel, there are hydrogen bonds between those cellulose molecules. The hydrogen bond forms between the hydrogen of one OH group, all right, the hydrogen of one OH group, and with the oxygen of another OH group. The cellulose molecules have overlapping ends, all right, which allows it to form into a microfibril. Part B. The width of stomatal opening in A. thaliana is regulated by movement of ions. These ions move through channel protein in the cell surface membrane of the guard cells. Draw a diagram to show part of the cell surface membrane with a channel protein, and then label the diagram. So guys, we have shown the phospholipid bilayer. We have shown the channel protein that is passing through both of the layers, and we have a hydrophilic core where the ions can pass through, water soluble ions can pass through, bypassing the phospholipid bilayer, which is hydrophobic, and hydrophilic heads and hydrophobic tails are labeled. Part 2, explain why channel proteins are needed for the movement of ions into and out of cells. You see, channel proteins allow facilitated diffusion. So facilitated diffusion allows ions, those that are charged, to pass through, with concentration gradient. All right, so cannot pass through the phospholipid bilayer because they are charged, so they will be able to pass through the phospholipid bilayer with the channel protein. Guys, if you have a better wording for this particular answer, go ahead with that particular point. However, in your wording, please keep that, channel protein allows facilitated diffusion, ions are charged, and because of that, they cannot pass through the phospholipid bilayer. As long as you have these particular points, that equates up to 3 marks. The question is for 2 marks. You can just, memorize this particular question. This is a pretty common question. And even if you get like 3 marks in your exam, you'll still make your marking point. Part C. The outward movement of ions from the guard cell causes stomata to close. A variety of A. thaliana does not have channel proteins for the outward movement of ions in the cell surface membrane of the guard cell. Suggest and explain the effect of not having these channel proteins on transpiration. So you see, if ions cannot move out, then the stomata will stay open. Transpiration rate will not decrease to a low level if the condition remains same. Water vapor will continue to diffuse out from the intracellular space because stomata cannot close. So the guard cells do not lose turgidity because there is no outward flow of water from the guard cell by osmosis because the ions did not move. So the water will remain within the cell. The plant is more likely to wilt. Question number 5. Measles is a highly infectious disease. The World Health Organization has coordinated a program for controlling this disease by using vaccination. A1. Name the pathogen that causes measles. Well, we know measles is caused by morbillivirus. Next, part 2. Explain how the measles pathogen is transmitted. We know that measles pathogen is transmitted as an airborne droplets. When an infected person coughs, sneezes, the airborne droplets are produced. When that airborne droplets are inhaled by an uninfected person, they can become infected. If a infected person sneezes, and then rubs that particular sneeze, and then they touch a surface, then that particular surface will then contain the virus. If an uninfected person touches the particular space and then puts their hand in the nose or in their mouth or in their eyes, then they can also get that particular virus. Part B. Figure 5.1 shows the number of cases of measles globally each month between January 2015 and March 2019. Okay. We can see that there is a trend as it goes from January, February, March, April. In April in 2015, that there is a, there is the highest amount. In March in 2016, we can see the highest amount of measles. In 2017, the amount of measles were less. The maximum amount occurred in March. All right. And in 2018, the amount of measles is less. The maximum amount occurred in, again, March. In 2019, the maximum amount can be seen in January. And out of all of this information, the lowest quantity is within 2019 in March. Okay. and the maximum is in 2019 in January. Describe the pattern of measles cases as shown in figure 5.1. We can see the measles cases throughout the year. First of all, it increases, then it decreases, then it increases and decreases back again. In 2015, January had around 2,700 cases. All right. And April has around, we can see 3,800 cases, and December has around 13,000 cases. The number of January 2019 are the highest. We can see in January 2019, it has the highest quantity, around 68,000. All right, cases, and March 2019 has also the lowest number of cases, which is around 3,000 or 4,000. Part C, explain why measles has not yet been eradicated, even though a vaccine has been available since 1960s. All right, those who are infected with measles can infect many people. And the person necessarily doesn't understand that they are infected with measles before they are actually spreading that virus. Measles spread to children before they are vaccinated. Vaccine cannot be given soon after birth because of passive immunity provided by the mother. Some people have poor response to vaccine when they are given the vaccine because of their poor diet and lack of protein in their diet. It is difficult to reach all people in terms of vaccination in the rural areas. And when the people from the unvaccinated people from the rural areas moves into urban areas, then they basically can spread the disease. Vaccination program can collapse as a result of war. Many children do not receive boosters. The vaccine administration requires trained professionals, which may be unavailable, which may be unavailable in rural areas. Question number 6. Table 6.1 compares some of the features of DNA with protein collagen. Complete table 6.1. Elements. In DNA, we know in DNA there will be carbon, there will be hydrogen, there will be oxygen, there will be nitrogen, there will be phosphorus as well. Monomers of DNA are nucleotides. So DNA nucleotides. And for collagen, it will be amino acids. Bond between the monomer will be phosphodiester. And in collagen is going to be peptide. Site of production in eukaryotic cells, it will be in DNA, and collagen will be in ribosomes. Guys, each point has 1 mark. Most of the DNA in eukaryotic cell is located in the nucleus. State how the structure of nucleus is suited to its function of containing DNA. We know nucleus contains membrane, forming, it has a nuclear envelope which has a double membrane. The nuclear pore allows small molecules, all right, to pass through. However, the DNA is too large to pass through, and it protects the DNA from hydrolytic enzymes that are available in the cytoplasm. Question part C. Fibroblast cells produce elastic fibers and collagen fibers in lung tissues. Describe the function of elastic fibers in lung tissue. Elastic fibers allows alveoli to stretch and recoil during inhalation and exhalation, which is very important because when the alveoli can recoil back to its smaller shape, it can ventilate most of the air, all right, which contains higher proportion of carbon dioxide. This prevents the alveoli from overstretching and bursting. Elastic fibers recoils to move air out of the alveoli. D. Name the stage of the cell cycle during which DNA and proteins are synthesized. So basically, DNA is synthesized in the S phase or the synthesis phase. So guys, that's all for today's question paper. We are solving questions, answers like this every day and uploading in our channel. So, subscribe to our channel and stay connected so that you get, answers for your question paper. And if you have any question papers of interest that you want us to solve, that has to be from chemistry and biology, let us know and we'll definitely solve it and we will upload it. Thank you very much for joining the class today. See you in the next class.